All right, so this chapter is talking about chromosomes and cellular reproduction. So we're going to go into chapter 2 in the genetics conceptual approach. In order for me to really help you understand or grasp the concepts of chromosomes and cellular reproduction, there's a couple things that we have to talk about, which should hopefully lay the foundation for those concepts. One, and that is your understanding of the difference between a prokaryotic cell and a eukaryotic cell, which would then further help you understand the difference between cell reproduction and sexual reproduction. So we're going to go into these concepts. So specifically a prokaryotic cell, okay? A prokaryotic cell is a unicellular organism which has what we would consider to be a relatively simple cellular structure, okay? That is different from a eukaryotic cell, which has a more complex and a compartmentalized cellular structure because it has intracellular, it has components that have multiple organelles inside that are also complex within themselves. So there's a vast difference between a eukaryotic cell and a prokaryotic cell. A couple of things to also look at is thinking about the organelles within the cell wall of a plant versus the plasma membrane of a eukaryotic cell. And both are plants and animal cells are considered eukaryotic cells, okay? From the perspective of genetics, a major difference between the prokaryotic cell and the eukaryotic cell is that the eukaryotic cell has a nuclear membrane which surrounds the genetic information, right? The DNA, the RNA, and it forms what we call the nucleus, okay? That separates the DNA from the other cellular content. We don't see that in a prokaryotic cell, okay? In a prokaryotic cell, the genetic material is in close contact with other compartments or components of the cell, but it is not a membrane-bound nucleus. Another fundamental difference between the prokaryotic cell and the eukaryotic cell is the packaging of their DNA. In eukaryotes, the DNA associates closely with what we call histone proteins, okay? These histone proteins help form the tightly packed chromosomes that we see, okay? A less condensed form of the DNA or chromosomes is called chromatin, and this is where you have the DNA that is interwoven or interacting with histone proteins, but they're not as condensed. Now, a couple of things to also keep in mind outside of the difference between the nucleus. The size of the cell of a eukaryotic cell is significantly different. A prokaryotic cell is relatively small in comparison to a eukaryotic cell. Okay. Another difference is the genome, which is the genetic information. In a prokaryotic cell, you typically have one circular DNA molecule that accounts for the cell's entirety, whereas a eukaryotic cell is going to have multiple linear DNA molecules. And then the amount of DNA that's present is significantly different. The amount of DNA in a prokaryotic cell is much smaller than that of a eukaryotic cell. And then you also have the membrane-bound organelles that I was just speaking about. You're going to have them present in a eukaryotic cell, but absent in a prokaryotic cell. So there are some clear differences between the two. Okay, so again, unicellular for a prokaryotic cell, no membrane-bound organelles, and made up of eubacteria and archaea. The eukaryotic cell has both unicellular and multicellular organelles. The genetic material is going to be surrounded around that nuclear envelope to form what we call a nucleus, and the DNA is linear, typically linear, okay, and is associated or tightly woven with other proteins such as histone proteins, which help form the discrete chromosomes that we see if we were to look under a microscope. Now, viruses are neither prokaryotic or eukaryotic because they don't possess the structure of a cell, okay? Viruses actually have a simple structure that is composed of an outer protein coat and that encapsulates the nucleic acid, whether it be DNA or RNA. All known viruses are also going to reproduce only within host cells. So they require a host cell to reproduce, and their evolutionary relationship to cellular organisms is really unclear. It's not very clear about their evolutionary relationship, but we do know that they are not considered living, okay? They're in the state between living and non-living. So with prokaryotic cell reproduction, okay, when a prokaryotic cell reproduces, its circular chromosome replicates. The cell divides in a process called binary fission, okay? Replication is usually going to begin at a specific place on the circular chromosome, which we call the origin of replication. And as a result, the rate of replication in a prokaryotic cell tends to be much higher than a eukaryotic cell. Okay, specifically under optimal conditions, bacteria cells can divide every 20 minutes. Okay, this is significantly different from a eukaryotic cell. The mere presence of multiple DNA molecules requires a much more complex mechanism to help ensure that only one copy of each DNA molecule is produced. Because if you have too much DNA in a cell, it can result in a genetic disorder and ultimately abnormalities. The nucleus in a eukaryotic cell has a highly organized internal, what we call scaffolding, okay, which is called the nuclear matrix. And this is composed of a host of protein fibers. The nuclear membrane also, or matrix, helps to maintain spatial relationship amongst compartments in the nucleus, and it's very important in DNA replication. We also see an association between the structure of the chromosomes in a eukaryotic cell with the cell cycle and potential consequences of the cell cycle. Okay. So what do I mean by homologous pairs? So in a cell, if we're talking about a diploid cell, di being two, okay, a diploid organism has two sets of chromosomes that are organized as what we call homologous pairs. So they're the same chromosome or the same type of chromosome, they just may have differences, as one would come from the maternal side and one from the paternal side, okay? But ultimately, we say humans have 23 pairs of chromosomes, or 46 chromosomes. On each chromosome, you're going to have various genes that are present. If you have a diploid organisms, then they'll have alternative forms of the gene, which we call alleles. Okay, this is different from a haploid cell because a haploid cell contains half of the genetic information, so they don't have homologous pairs of chromosomes. They only have one set of a chromosome for each specific chromosome. They don't have double the amount of genetic information. So looking at the chromosome structure, there are what we call three main components of the chromosome structure. You have the centromere, which is the attachment point for the spindle microtubules. That is the restriction point on the chromosome. Now, I won't say that it's always going to be in the middle, because depending on the type of chromosome, the centromere can be present throughout different portions of the chromosome. However, we do know that it is a restricted point of the chromosome, and it typically tends to be repetitive DNA. Okay. The telomeres are also a part or a component of the chromosome, and this is the ends or the tips of a linear chromosome, and they also are repetitive sequences. Okay, these repetitive sequences are actually thought to help stabilize the ends of the chromosome so that they don't shorten through each round of replication, and they stabilize it and provide stability to the chromosomes. Interestingly, research shows that telomeres also prevent in limiting cell division, and they play an important role in aging and cancer. One of the important aspects that we'll talk about in chapter 12 is understanding that component, is especially associated with cancer. One of the hallmark findings in cancer was that all cancer cells have an abnormality in their telomeres, and that helps them to be immortal or to continue to grow. Hence, the concept of the HeLa cells, or the Henrietta Lacks cells, which are considered immortal, and it's been shown that they have abnormal telomerase activity. Telomerase is the enzyme that helps extend out the telomeres of chromosomes. And then you have the origin of replication, and this is where DNA synthesis or DNA replication begins, okay? Unlike centromeres and telomeres, they're not easily observed in the microscope. Okay, their structure and function will be discussed further in the as we move throughout the semester. But their structure and function is not fully observed under a microscope like you would see like telomeres. In preparation for cell division, each chromosome replicates and it makes a copy of itself. The two initial identical copies that are made are called sister chromatids. And that is what is held together at the centromere. So when the cell undergoes DNA replication, it makes a copy of that chromosome of the DNA, and having two copies of the chromosome are called sister chromatids. Okay, each sister chromatid consists of a single molecule of DNA. So looking at the structure of the chromosome, you'll see that eukaryotic chromosomes can be divided into four major types based on the positioning of the centromere. Okay, based on the positioning of the centromere. And one thing that I want to make sure I point out, let me back up for a second to explain this portion. The centromere is also the docking site to where the connector core, which is a disc-like protein structure, binds to and is very essential in the process of cellular division. Okay. This is where the spindle microtubules will actually bind to and begin the process of the pulling apart, or putting that force that pulls apart or separates the sister chromatids from each other. Okay, so this is very important. So back to the chromosome structure, you have four major types: submetacentric, metacentric, telocentric, and acrocentric. To give you an idea or a succinct explanation of them, how to identify the differencing of the chromosome structure is look at the placement of the centromere. The placement of the centromere is here. Okay, look at the placement of the centromere, and you'll see that the placement of the centromere differs for each type. Here we have one that actually is about a fourth of the way from the top. Okay, actually about 2/4 of the way from the top. Let's say a third of the way from the top, and we call this sub-metacentric. Okay, metacentric, think about it as meat, so, or middle. The centromere is in the middle of the chromosome. Okay, so it divides it in two equal halves. Telocentric is where the centromere has terminalized to the end of the chromosome, so it does not have two equal parts or even two equal parts. The telomere, the centromere is on the end of the chromosome. And then we have acrocentric. Acrocentric is where you have just a little bulb on the end of the chromosome, because the centromere is towards the end of the chromosome. Okay, so you have the small little piece of the chromosome on one end, and then a larger portion on the opposite end, which sometimes can be referred to as stalk and bulb. But ultimately, the placement of the centromere allows us to distinguish the one chromosome from another, or how we label the chromosomes based off of the presence of the centromere. So let's go into the cell cycle. Okay, the cell cycle is a series of stages that the cell goes through or passes through when it goes from one division to the next. And it is through the cell cycle that the genetic instructions, so the DNA, okay, for all characteristics are accurately passed from parent to daughter. So this is how we get the passing on of genetic information from one cell to the next. Okay. Once the cell cycle is occurring, you have this an active cell that is dividing. Okay, you'll start to ultimately, the end goal of the cell cycle is to produce at least 2 new cells. If we're talking about the basis of what we call mitosis, okay, is to produce 2 new cells. Okay, at the end of that cycle, the cell divides to produce 2 cells, which can undergo additional cell cycle rounds of cell cycle. However, the process of the cell cycle is tightly regulated, so much so that there are what we call checkpoints. So before the cell can go through these different phases or stages in the cell cycle, there are transition points that are required for the cell to undergo to ensure that there are no damaged or abnormalities present. These checkpoints are necessary to ensure that all cellular components are present and in good working condition. This helps to prevent damaged cells, abnormal cells, from passing on their genetic information to a new cell. The cell cycle we can divide it into 2 major phases. The first is interphase. This is the period where in between the actual division. So this accounts for cell growth, development, and function. And it's critical for cell division to take place. It's critical. It's one of the most critical events. The second is the M phase, or the mitotic phase. This is where you have a period of active cellular division. Okay. And then you have cytokinesis. Okay. Cytokinesis is the portion where you have the cytoplasm that is divided. So mitosis is where you have the genetic information or the nuclear division, and cytokinesis is where you have the cytoplasm that is divided. So let's look into cell cycle. So you'll see here, the first part of the cell cycle is called the G1 phase. Okay, this is the phase where the cell is growing, but the cell is not just growing. The cell is also producing all of the proteins and the constituents needed to carry out the upcoming phase, which would be the S phase. So you have the G1 phase where the cell is growing. It's producing all of the constituents, the components needed to carry out the S phase. What is the S phase? The S phase is DNA replication, synthesis of DNA. Okay. But before the cell can go into the S phase, the cell has to pass this checkpoint, this transitionary point. This G1 S checkpoint is present so that the cell can ensure that there's nothing wrong. Okay, that everything that is needed is present. All the proteins that are needed are present. There's no damage to the cell, and that prevents it from going into the S phase and duplicating abnormal DNA. Once the cell has committed to dividing and it's successfully passed through that first G1S checkpoint, it goes into the S phase where it undergoes replication in a process we know as DNA replication, which we'll talk about. After it goes through DNA replication, it proceeds to G2. Okay, G2 is the next phase, and G stands for gap. Okay, the gap phase, where also is growing. Okay, the cell surface area has significantly increased because now it has double the genetic information, but it now has to provide double all of the constituents and components within the cell so that it can divide into 2 equal cells. So that means the cytoplasmic portion is going to increase. The organelles are going to be increased so that you can have double the genetic information. Okay. Once it goes or finishes G2 cell, the G2 phase, which is preparing for mitosis, it has to go through a point which is called another checkpoint called the G2m checkpoint. And this is right before it's able to go into mitosis. And this is important because the cell does not want to allow a cell to go through mitosis and produce another cell or 2 new cells that are abnormal. So if the cell does not have all of the components needed to go through mitosis, it will be marked for death, or it will be halted and not allowed to progress into the mitosis or the M phase. The M phase, which is the phase where the cell begins to undergo mitosis, which is the division of the nucleus, and then the cytokinesis, which is the division of the cytoplasm. This is the overall summary of the cell cycle. We're going to go through these components. So first, with a interphase, as I kind of explained, these are the components of interphase: the G1, G2, and the S phase, and also G sub O. Okay, the G sub O phase, a cell can remain in this quiescent phase for an extended period of time, and it's really cell dependent. Okay, it is really, really cell dependent. But that G sub zero phase will allow for a cell to remain there if maybe something is wrong or it's not ready for it to divide. So it needs to be a non-dividing cell. It'll stay at that stable state for a period of time so that the information, the genetic information, stays constant. So let's go into the the M phase. Okay, this is the phase of the cell cycle where copies of the cell's chromosomes, which is the sister chromatids, okay, separate and the cell undergoes division. So I want you to keep in mind what we just talked about. The M phase cannot occur if the S phase doesn't occur. Okay, DNA replication is essential for the M phase because going into the M phase, one can assume that all the genetic information has doubled. Okay, if the cell doesn't go through the S phase, then the M phase cannot occur because what we'll have is an abnormal amount of genetic information. Okay, so in the M phase, the separation of the sister chromatids is a critical process that results in a complete set of genetic information for the resulting cells that are produced at the end of mitosis. Okay, mitosis is a continuous process, and it is separated into what we would consider like arbitrary 6 steps or 6 phases. So depending on the textbook you may read, it may say 5. Okay. Let's go to the first phase. So the first phase we have is prophase. Okay. Oops, let me go back. Prophase. In the process of prophase, and I'll show you some diagrams in a second, but you have some key components, which there's also a table provided for you. You have the disintegration of the nuclear membrane. I'm sorry, let me back up. That's prometaphase, but prophase is where you have the chromosomes starting to condense. Okay, prior to prophase, the chromosomes in the cell are not identifiable. You can't distinguish one from another. Okay, so as the cell enters prophase, the chromosomes are beginning to condense so that they can be discretely identified one from another. Remember, it's setting itself up to divide the cell. So in order to efficiently and effectively divide the chromosomes, they have to be distinguished one from another. Okay, so this allows the cell's prophase to become more compact and visible under a light microscope. One of the key components to helping the chromosomes condense, okay, or the DNA condense, the chromatin condense to chromosomes, is proteins called condensins. And these proteins bind to the DNA within the chromosomes and they bring about condensation, okay, of the DNA. This is extremely important. Again, the cells have to be able to identify it or discretely identify the chromosomes from each other. Okay. The next is prometaphase, or sometimes you may hear it as late prophase. Okay. But in prometaphase, you start to have the disintegration of the nuclear membrane. So that's one thing to look at. If you're looking at cells under the microscope, pay attention to some of these key components to identify what stage of the cell cycle of mitosis the cell is in. If you still see a nuclear membrane, then 9 times out of 10, you're still in prophase. Okay. In prometaphase is where the nuclear membrane begins to disintegrate and the spindle microtubules, which are outside of the nucleus, are now moving into the nuclear region to help with the process of dividing the chromosomes. Eventually, during prometaphase, the chromosome is going to become attached to the microtubule from opposite ends of the poles, which we consider each area of the cell a different pole. And the microtubules from one of the centromeres, the microtubules are going to connect to the connector cores that are bound to the centromeres that we previously talked about. Metaphase is the process where the cells meet at the middle or the equatorial plane of the cell. And I'm going to show you some diagrams in a second. But in this case, the chromosomes become arranged in a single plane between the two centrosomes. Okay, the centrosomes, not centromeres, the centrosomes are now at opposite ends of the cell with an irradiating, okay, or radiating from the centrosomes are the microtubules that are going towards the chromosomes, specifically the connector cores. Tension, because of that, tension is starting to build up at the connector cores that are joining the two chromatids together, and this is going to help with the pulling apart of the sister chromatids. This tension is required for the pulling apart, okay, or for the cell to pass through the spindle checkpoint. This is required. The next phase is anaphase, okay, and in anaphase, after the spindle assembly checkpoint has passed, the connection between the sister chromatids breaks down, okay, and the sister chromatids are now separated. When you see the separation of the sister chromatids, it marks the beginning of anaphase. And this is where the sister chromatids, or the chromosomes, I'm sorry, at this point are going to move to opposite poles. Okay. So at this point in anaphase, you should not have the sister chromatids because they have now been separated. Okay. Which brings us to telophase. In telophase, the sister chromatids have separated and they're now considered a separate chromosome. Telophase is marked by the arrival of the chromosomes to the spindle pole, so to each ends of the cell. And now the nuclear membrane is going to begin to reform around the new set of chromosomes that are present. This is also setting up for the division of the cytoplasm. There's an animation in the book. If you look on around page 26 or so, there is a link that you'll be able to click on if you have the ebook to look at a nice animation of it. It's animation 2.1. A key component that I want to talk about, because we've been talking about these spindle microtubules, okay, that are very essential in the pulling apart or forming that tension that helps to separate the sister chromatids from each other at the centromeric region where it is where there's a restriction or a pinch that has the connector cores present. The spindle microtubules are made up of what we call tubulin subunits, okay, and each microtubule has a plus end and a negative end. The plus end at the connector core and the negative end at the centromere. So there's two different portions. The positive end of the tubulin is going to be closer to or attached to the connector core, and the negative end is going to be towards the centrosome. Okay, the negative end towards the centrosome. So this also is allowing you to see that the positive versus negative role has a great effect on the tension that is actually building to help with the separation of the sister chromatids. Utilize this table 2.1 to really understand a succinct explanation for what is happening at each of these phases. And one that I want to mention is cytokinesis. And I've talked about this already, but cytokinesis is the cytoplasm division. So this is where the cytoplasm of the cell divides. From prophase to telophase, we had nuclear division. So the nucleus, the contents within the nucleus are now divided. That's the nucleic acid. Cytokinesis is where the cytoplasm now divides so that it can form two equal cells. Okay, in a plant cell, it is the cell wall that forms. So what are some genetic consequences of the cell cycle? Okay. One, know that the cell cycle is producing two cells that are genetically identical to each other. And with that, with the cell, it gives rise to them. Okay, it gives rise to new cells. You also have newly formed cells that contain a full complement of chromosomes. So one cell is going to produce another cell, a sister cell or daughter cell, that has the full complement of genetic information in that cell. Okay. An important genetic result of the cell cycle is that each of the cells produce a full complement of the chromosomes. There's no reduction or increase in chromosome number. It stays the same. Each cell also contains approximately half the cytoplasm and organelle content of the original parent cell, but no precise mechanism. Remember, we talked about those stages of the cell cycle, specifically interphase. During interphase is where you have the doubling of the content of the cell so that it can produce two new cells, okay, or one cell buds off from it. So this is why you have that information that is, or a new daughter cell that is able to be derived from that parent cell and give half of the cytoplasmic portion. So if you were to look at a dividing cell, an active dividing cell is going to be larger in the surface area than a non-dividing cell. Okay, the number of chromosomes and the number of DNA molecule can change throughout the course of the cell cycle. Okay, the number of chromosomes per cell equals the number of functional centromeres in a cell. The number of DNA molecules per cell equals the number of chromosomes when the chromosomes are unreplicated. Okay, and twice the number of chromosomes when the sister chromatids are present. So this table will really help you understand the number of chromosomes in the cell and the number of DNA molecules in the cell based off of the state of the cell cycle. I would definitely give that table further exploration. So let's go into sexual reproduction and genetic variation. Okay. If all reproduction was accomplished through mitosis, life would be dull. It would be boring because remember, mitosis produces genetically identical progeny, produces genetically identical cells. So there is a benefit to having sexual reproduction because without sexual reproduction, everything would be the same. Okay, everything would be the same. Only the occasional mutation would introduce any genetic variation. All organisms reproduce in this way for the first 2 billion years of the Earth's existence. Okay, and it's believed that there was an evolutionary transition of reproduction. Specifically about 1.5 billion to 2 billion years ago, something remarkably is thought to have evolved, and that's cells that produce genetically variable offspring through sexual reproduction. The evolution of sexual reproduction is the most significant event in the history of life. It consists of two processes, okay? The first process is meiosis, or meiosis, and that is the production of haploid gametes. Remember, we talked about haploid and diploid. Diploid is an organism that has two copies of genetic information. Haploid has half, or only has one copy of the genetic information. Okay, so meiosis is going to lead to gametes in which are sex cells, which have half the number of genetic information. And then the second process is fertilization. And this is which you have two haploid gametes that fuse and restore the number of chromosomes to its original diploid value. So meiosis is the production of the haploid gametes. Fertilization is the fusion of the haploid gametes. Now, what's important about sexual reproduction is that through meiosis, one of the processes of sexual reproduction is meiosis, and it gives genetic variation. This is how we prevent having identical organisms or species. So let's go into meiosis, okay? Don't confuse meiosis with mitosis because they're not the same thing, okay? Mitosis differs from meiosis in several ways. One, mitosis consists of a singular or a single nuclear division and is usually accompanied by a single cell division. Meiosis consists of two divisions. So it has two rounds of division, okay? After mitosis, the chromosome number of the newly formed cells is the same as the original cell, whereas meiosis consists of the chromosome numbers are reduced by half. Another difference is mitosis produces genetically identical cells and meiosis produces genetically variable cells. All right, so let's look at the first division of meiosis, which is termed meiosis I. And in meiosis I, you have the separation of homologous chromosome pairs and the reduction of the chromosome number by half. The second of division, which comes after meiosis II, is termed the equational division. The events of meiosis II are similar to those of mitosis. So when you look at meiosis II, you can kind of think of it as synonymous with mitosis. However, it differs from mitosis in that chromosome numbers have already been halved in meiosis I, so the cell does not begin with the same number of chromosomes as it does in mitosis. So let's go into prophase I. And I like to tell students one of the most important phases of meiosis that make it significant and significantly different from mitosis is prophase I. A lot of the key components that are unique to meiosis happen in prophase I. Okay, so in prophase I, it is a pretty lengthy stage, so much so that it's divided into 5 substages, okay, which we're going to talk about in a second. But you have a couple of key components or things that happen here. In prophase I, you have a synapsis that forms, and it's like a zipper-like structure that helps to close the pairing of the homologous chromosomes. So it brings the homologous chromosomes together in what we call a synaptical complex. It's a zipper, it looks like a zipper-like structure, and it brings the 2 homologous chromosomes pairs together so that it can prepare for crossing over. We also have what we call the tetrad. Okay, each homologous pair of synapse chromosomes is called what we call a bivalent or tetrad, which consists of 4 chromatids. Okay, 4 chromatids. And then we have crossing over, as you've heard me mention, crossing over of chromosomes, segments from the sister chromatid of one chromosome to the sister chromatid of the other synapse chromosome. So another way of saying that is when you want to have the exchange, the physical exchange of genetic information, it is crossing over that helps generate that genetic variation through the physical exchange. And it is essential for the proper alignment and separation of the homologous chromosomes. Okay, each location where the 2 chromosomes cross is what we call the chiasma. So let's go into looking at these substages, which is significant for crossing over. This is how crossing over occurs. So in the first stage, you have leptotene. You may hear it as leptonema. The chromosomes in leptonene condense and become visible. In zygotene or zygonema, the chromosomes continue to condense and homologous pairs pair up and begin to form that synapsis or synaptical complex. The next you have pachytene, okay? And in pachytene or pachynemia, you have the synaptical complex that has actually formed, and you have the moving or the preparation of the crossing over to form. The next is diplotene or diplonema. This is where the centromeres of the pair chromosomes move apart, but the 2 homologous remain attached at the chiasma. That's the place where crossing over occurs. And then you have diakonesis. This is the final substage of prophase I. And in this phase, chromosomes begin to condense even further, and the nuclear membrane breaks down. The spindle fibers begin to set the stage for metaphase I. So these are the substages of prophase I, and these are all the stages that break up the process of crossing over, which are key and begin between zygotene and diplotene. So in metaphase I, you have the random alignment of homologous pairs of chromosomes along the metaphasic plate or the middle of the cell. And it is initiated when the homologous pairs align themselves. Anaphase is marked by the separation of the homologous chromosomes and the random distribution of chromosomes into 2 newly divided cells. This is the second mechanism of generating genetic variation in newly formed gametes, because how they align or line up at this cell can produce a different variation. Then we have telophase 1. This is where the chromosomes arrive at the spindle poles and the cytoplasm divides. Okay, the cytoplasm divides. All right, so now we're going into Meiosis II. Meiosis II is the second round of division. Now, one thing I want to point out, and as you'll see here in this last one, you have interkinesis, okay? The period between Meiosis I and Meiosis II is called interkinesis. And this is where the nuclear membrane reforms around the chromosomes clustered at each pole, the spindle fibers begin to break down, and the chromosomes relax, okay? This will then allow for the cells to go through and Meiosis II, which will be started by Prophase II, and the processes or the stages are the same. They just have a 2 at the end. So it's Prophase II, Metaphase II, Anaphase II, and Telophase II. The processes in this portion are similar to that of mitosis. I'll just kind of give you some key components or steps that may help, and you can also use the diagram at the bottom, these to help you, okay? In Prophase II, the processes or the events of interkinesis are reversed. The chromosomes recondense, okay? The spindle fibers reform, and the nuclear membrane breaks down again. In Metaphase II, which is similar to the metaphase in mitosis, the replicated chromosomes line up at the metaphasic plate in the middle of the cell, with the sister chromatids facing opposite poles. In Anaphase II, the sister chromatids separate, and the chromatids are now pulled to opposite poles. Remember, in Anaphase now, we no longer have the sister chromatids anymore. It is at Anaphase II that each chromatid is now a distinct chromosome, so no more sister chromatids. In Telophase II, the chromosomes arrive at the spindle poles, and a nuclear membrane is going to begin to reform in Telophase II around the chromosomes. And now it's going to prepare for the division of the cytoplasm. The chromosomes are relaxed and are no longer visible. So you have this cycling of condensing and decondensing throughout meiosis. This table is going to, I've again provided all of the major features in Meiosis I and Meiosis II. So what are some consequences of meiosis and genetic variation? Okay. 1, 4 cells are produced from each original cell. Prior to a difference between mitosis and meiosis, mitosis produces 2 genetically identical cells. Meiosis produces 4 cells that are each different. They're each unique. You also have the chromosome numbers in the cell that are reduced by half. So they're not diploid, they're haploid. Okay, these are, again, sex cells that we're talking about, gametes. And the newly formed cells from meiosis are genetically different. As I stated, they're not the same. Even from the parental cell, they're not the same. Okay, so these are what we would consider to be consequences of meiosis and genetic variation. Okay, in this example that you're seeing here, the cell possesses 3 homologous pairs of chromosomes. However, you'll see here the cell has 3 homologous pairs of chromosomes. Okay, 1, 2, 3. This is one set. This is the other set. Okay, each pair is maternal in origin, and the other is paternal. So here is the maternal in red. The paternal is in blue. Okay. There are 4 possible ways to align at the metaphasic plates, as you see here. This is one way that gives genetic variation. How they align at the metaphasic plate is providing levels of genetic variation. And in this case, there are 8 different combinations of chromosomes in the gametes that are possible. And it depends on how they align and separate in Meiosis I and Meiosis II. Okay, so this is another way or manner by which you can get genetic variation just by merely lining up at the metaphasic plate. And this is different from crossing over. That's another level of genetic variation. Okay, so when you look at the comparison or the concept of mitosis versus meiosis, you'll see that there are some stark differences, okay? Very stark differences between the two. All right, one, notice here, mitosis, you have the parental cell. It's diploid. It goes through prophase, metaphase, anaphase, and it produces two diploid daughter cells that are identical. Okay, you can even look at the genetic information to see that they are genetically identical. However, in meiosis, you have a diploid parental cell. It goes through the first round of meiosis 1. You know that because after prophase, metaphase, anaphase, there's a 1. So it lets you know that it goes through meiosis 1. Here, you'll see that in prophase 1, that's the place where crossing over takes place. And you can see that the chromosomes are actually crossing over. We see the presence of that chiasma. Okay. In metaphase, we see that it is aligned at the metaphasic plate, which is another level of genetic variation. So we get that genetic variation in prophase 1 and metaphase 1. You have the pulling apart anaphase, then you go through the telophase, and then you have the interkinesis, okay? Metaphase goes through a meiosis 2, but the end of meiosis, okay, that's 1 and 2, the end of meiosis results in 4 daughter cells that are now haploid, and each of them are genetically unique. They're all different, and you can just look at the chromosomes to help you understand that visually, that they are different. Okay, this table here should be of help as you're studying to understand the comparison of mitosis, meiosis 1 and meiosis 2 and how they are in some instances similar. Okay, specifically meiosis 2 and mitosis. So a couple other things that I want to point out to you is the separation of the sister chromatids and the homologous chromosomes. One of the key factors or components here that are important is cohesin. Cohesin is a protein that helps the chromatids to be held together, and it is a key to the behavior of the chromosomes in mitosis and meiosis. Okay, these cohesin helps to is present along the chromosome arms, and it holds the homologs together at the chiasma. Okay, so it helps hold them together. This is very, very important because if you did not have them holding if the cohesin was not present, you would have the breaking apart of the chromosomes, and it would make it much more difficult for them to be held together. So let's briefly go into meiosis in the life cycle of animals and plants. Okay, meiosis in animals for the males is called spermatogenesis, and for females it is called gametes. It's called oogenesis. So female gamete production or egg is considered oogenesis or termed oogenesis, and in males it's called spermatogenesis. Okay, and we'll also briefly go into the process in plants. Couple of things that I want to point out for spermatogenesis and male formation, gametogenesis, is specifically when you're talking about the formation of sperm, okay, because that's what spermatogenesis is, the formation of sperm. It starts off with spermatogonia in the testes, and it undergoes repeated rounds of mitosis to produce more spermatogonia. The spermatogonium will enter into prophase 1 and become what we call a primary spermatocyte. The primary spermatocyte then goes through meiosis, okay? And it will produce 2 secondary spermatocytes. It'll then go through, finish undergoing meiosis. And at the end of meiosis, you'll have what we call 4 spermatids, which will then mature to form a tail, or had on the addition of a tail, which are then termed sperm. In female gametogenesis or oogenesis, the oogonia in the ovaries may either undergo repeated rounds of mitosis to produce additional oogonia, but eventually the oogonium will enter or form what we call primary oocytes. The primary oocytes will enter into meiosis I, as you see here. Okay, it'll enter meiosis I, and at the end of that, of meiosis I, you'll have two cells. One is the primary oocyte. One is the what we call polar body. Okay, so you have unequal distribution of the cytoplasm, to where one is going to eventually disintegrate the polar body because it is not a viable cell. It'll go through the second round of meiosis. And at the end of meiosis, you're going to produce one ovum and 3 polar bodies. Ultimately, what you have is only one viable cell that can undergo the process of sexual reproduction in comparison to the males, which produce 4 viable cells. Okay, so the end of oogenesis for a female is only going to produce one viable egg cell. The males will still produce the 4. Each are still going to be genetically different or unique. During the process of fertilization is where you have the fusion of egg and sperm. Okay, so the fusion of egg and sperm. Okay, the fusion of this egg and sperm will produce a zygote. That process is called fertilization. Okay, now in plants, that process is a little bit different. Okay, because meiosis in plants is a little bit more, it's similar in terms of the male versus the female concept, but it also is a little bit different. Okay, you have the male and the female part of the plant. Okay, the ovary or the pistil is the female portion of the plant, and that is called the megasporocyte. The male portion is called is where we call the stamen. Okay, and that's microsporocyte. It will undergo mitosis and meiosis. At the end, however, you're going to still produce 4 haploid cells, and then the end of, for the female or the megasporogenesis, it's going to produce 1 egg that is haploid, and you'll have the fusion, as you see here. Okay, the fusion of the egg and the sperm to begin that process. Okay, so that is the end of chapter 2, going through chromosomes and cellular reproduction. I want to strongly suggest that you go through the book even more to read. In this class, you have to have the textbook. You can use the ebook. I suggest the ebook because you can highlight, you can, there's a digital notebook there to help you study. And so definitely go through. I can't touch everything in the PowerPoint, but this is just the highlighted points so that you can go back through and read through on your own to have a better understanding of the concepts more in depth. Throughout the textbook, as you're going through it, you'll see that there are more animations that you also can view to help you understand these concepts.